lymphatic cells. And the lymphatic cells are those cells you find abundantly throughout the lymphatic system. Now, this is not to be confused with the lymphocytes, because lymphocytes are just a sort of a subcategory here. Lymphatic cells are just the ones you're going to find more commonly in the lymphatic system. So this includes things like macrophages, nerve cells, dendritic cells, and lymphocytes. Macrophages are a type of cell that they're a part of your innate immune system, and they just can recognize something that's foreign and then try to engulf and digest that through phagocytosis. Now, dendritic cells are very similar to macrophages, but dendritic cells are more mobile. In fact, dendritic cells, another word for this is the Langerhans. You guys remember that one from skin? So you still find these dendritic or Langerhans cells in the lymphatic system, not just skin. But it makes sense. You find a lot of lymph vessels in the skin too. Now, these dendritic cells are like a mobile macrophage. They can move around more often, and they can actually kind of scavenge around tissue. And they also look for things that are foreign. But they're also part of your innate immune system. So they just identify things that are foreign and then remove those from the body. Not necessarily like a specific antigen, but things that are have different antigens than your own body cells. Nerve cells are a type of cell that help modulate immune system activity, and then lymphocytes include several subcategories of cells that we'll talk about. In fact, for the lymphocytes, we got T lymphocytes, B lymphocytes, and natural killer cells. So the T lymphocytes are ones that can specifically fight infection. We'll talk about how that works. And there's actually subcategories of T lymphocytes as well. The B lymphocytes are the antibody secreting cells. So remember we talked about with blood typing how you can make antibodies against a foreign antigen. Like if you have type O blood and you're exposed to blood with A antigen on it, like you're going to make antibodies against that particular antigen. So these B lymphocytes actually can make antibodies to help fight off infection. But what's important to note about these B lymphocytes, you guys, is they respond to one specific type of antigen. Okay, so that each B cell secretes its own type of antibody that specifically targets one type of antigen. So what this means then is that if you are like type O blood, and you receive an improper transfusion from like type A blood, then you're going to make B lymphocytes that only make anti-A antigen or anti-A antibodies. And that specific cell will only make an antibody against that one antigen. Okay? So they are very specific in terms of their immune response, which can be good and bad, and we'll come back to this. But the thing about these T cells and B cells, you guys, is these are part of your learned or adaptive immune response. In fact, it takes time and exposure to form a memory using these T and B cells. So you need T cells and B cells here to be exposed to an antigen before they can even make the antibody against that antigen. Okay? Natural killer cells, though, these are actually part of your these are part of your innate immune response too. And they help initiate things like apoptosis in foreign cells. We'll come back to that. Now, for the T lymphocytes, they're the most abundant of your lymphocytes. So they make about 70 to 85% of your body's lymphocytes. In fact, how abundant were lymphocytes in terms of all the leukocytes in your bloodstream? What was our mnemonic? Never let monkeys eat bananas, right? So let was the lymphocyte. So these are the second most abundant of the leukocytes. But of those lymphocytes, most of those are the T lymphocytes. Okay? So that means that these are pretty abundant as a proportion of all your leukocytes in your bloodstream. And they express these co-receptors called CD receptors. And these CD receptors can help recognize a particular antigen. Can you guys give me an example of an antigen?
Blood type.
Yeah, blood type. So what's an antigen blood type? Yeah, A antigen or B antigen or D or RH. Good. So are you going to make antibodies? Are you going to fight antigens that your body normally makes itself? No, not under normal circumstances. So these CD receptors, these co-receptors, they're used to help identify self from non-self, and so that they can help recognize whether the cell belongs and it has normal antigens, or if it has antigens that aren't normally found in your body, they can help fight off that infection, okay, or at least remove those antigens. So there's two subclasses of the T lymphocytes. We got the helper T and the cytotoxic T. The helper T lymphocytes release cytokines, and cytokines are part of your inflammatory response. But think of these cytokines too, you guys, as like an alarm signal for infection. Because once you get inflammation due to helper T lymphocyte activation and cytokine release, those cytokines help excite other immune cells. So they kind of get the whole process going. Cytotoxic T lymphocytes, on the other hand, are the ones that don't just excite other cells. They actually help fight the infection directly. So for the helper T lymphocytes, you guys, these are CD4 positive. CD4 is a type of CD receptor you find on these particular T lymphocytes. And CD4 receptors are the ones that HIV uses to get into cells. So human immunodeficiency virus, HIV, attacks cells that are CD4 positive. Okay? So HIV then uses this CD4 receptor to get into the host cell. Once inside the host cell, then HIV can inject its genome into the host cell genome, like it becomes part of the cell's genome, and then it kind of hides there. So it's difficult for your immune system to find it because now it's part of your DNA. So the function of T helper cells, though, is they help with the immune system because they release cytokines when they recognize a foreign antigen. So let's say if you were exposed to a blood type that had an antigen that you didn't normally have, right? But let's say if you were like O negative, but then you got a transfusion from somebody who is like O positive, right? Well, that D antigen then would be considered foreign, right? It's not normally found in your body. So then what would happen then is these T helper cells could recognize that D antigen as being foreign, and once they recognize that D antigen is foreign, then they release a lot of cytokines, okay? And then these cytokines are involved in kind of just exciting the whole immune system. So they get the whole process going. So what does this mean then for HIV? Like if HIV infects CD4 positive cells, and CD4 positive cells release cytokines, what do you guys think is going to happen with like HIV?
Release cytokines.
You have difficulty releasing cytokines, right? Because that's the type of cell it's attacking and killing, right? And if you have difficulty releasing cytokines, what are you then not doing?
You're not sending the signal that there's an infection.
You got it. You're not exciting other immune cells. Yeah, you got it. I like that too. It's sort of like, what's that guy's name? Paul Revere or something? Yeah. It's like I like that too, yeah. The T helper cell is like the Paul Revere of your immune system, right? Like the British are coming, right? I don't think you had a British accent, you know what I'm saying, right? And so HIV, they would be like duct tape over his mouth, like, don't say a word. So then your immune system can't really get excited because then you're not releasing as many, it's not being stimulated by as many cytokines, okay? Now cytokines aren't only involved with stimulating other immune cells, they're also part of your normal inflammatory response, but inflammation helps set up the stage for tissue repair. We'll come back to that though in pathophysiology. Now that differs from the cytotoxic T cells. Cytotoxic, cyto means cell, right? Cytotoxic, which basically means it's kind of toxic to cells. So cytotoxic T cells are the ones that will specifically fight infection directly. How does that differ from the CD4 positive T helper cells? Are they fighting infection directly?
No.
They're alerted, yeah, exactly. Like they're sentinels, right? They send out a signal when there's an infection or a foreign antigen. But they're not participating in the immune response directly. They're just sort of exciting other cells. Now, those cytokines can go on and activate other immune cells like the cytotoxic T cells. And what these cytotoxic T cells do is they actually help initiate apoptosis in foreign cells. And so apoptosis is actually programmed cell death. And so when these when these CD8 positive cytotoxic T cells encounter a foreign cell and they bind to it, they basically tell it to kill itself. It's like the Grim Reaper of your cells, right? So these CD8 positive cytotoxic T cells can initiate apoptosis, and they basically cause these cells to kill themselves, which is actually pretty cool. So I like to think of it as like the Grim Reaper, right? And is that a thing like where you get touched by the Grim Reaper and apparently that's what kills you?
Yeah.
Like it just touches you, right? Yeah. So that's kind of like this. These cytotoxic T cells, CD8 positive, when they bind to a foreign cell, boop, they initiate apoptosis, and apoptosis is programmed cell death. So they kill off the infection directly that way. So what this is showing here, you guys, are the T helper cells. So T helper cells are involved with releasing cytokines when they recognize a foreign cell by recognizing its antigen. In fact, what does it use to recognize a foreign antigen? How do we know that what does a T helper cell use to recognize that this foreign cell is actually foreign? Yeah, CD4 receptor. You got it. So these CD4 receptors, if they can bind and sort of not really recognize, but when they bind and there's a chemical reaction that tells you that this is not a normal antigen in your body, what happens then is that this T helper cell then becomes excited. It activates. Now, when it activates, it proliferates into what we call clones. So basically, it starts causing itself to divide a lot, right? And as it's dividing and making more clones of itself, right, copies, each copy starts releasing a lot of cytokines. And cytokines were involved with
Drawing everyone else to the party.
inflammatory response, but they release cytokines. So cytokines are doing what? Other immune cells. You got it. They're activating other immune cells and helping to initiate certain aspects of the inflammatory response. So what these cytokines can do then is stimulate B cells, it can stimulate Treg cells, as well as stimulate the formation of macrophages. What was the function of a macrophage again? It's a big eater, right? So it's an engulfer, right? Now, B cells were the antibody factories, so that in response to these cytokines, you can start to activate the cells that pump out antibodies. And when they release these antibodies, those antibodies then can bind to the foreign antigen. And then those antibodies do other things too. So you can see here that there's kind of this interesting interconnected web of an immune response. We're starting it here, though, with the T helper cell. So what type of receptor would you find on T helper cells? CD4, and that's significant because HIV, you got it. HIV uses CD4 to get into those particular cells. So HIV only infects CD4 positive cells. Another example of CD4 positive cell, you guys, a dendritic cell is CD4 positive, and neurons. So later on when we get to pathophysiology, we'll talk about how chronic HIV infection also causes a type of dementia because HIV infects your neurons as well. So it's kind of interesting. What about dendritic cells? What were their function? Same thing as the macrophages, but they're very mobile, right? And this is actually how HIV spreads from the site of infection. Like, let's say if you got it like sexually, well, how does a virus, like in tissues associated with like sexual organs, how does it get to other parts of your body? Dendritic cells. Because once the HIV gets in a dendritic cell, and dendritic cells are mobile, they start spreading around your body, then the little virions can start to bud out of the dendritic cell and then infect other nearby cells. It's like a Trojan horse kind of thing. So, yeah, no pun intended, right? Should have used a Trojan to prevent that. Actually, we'll learn, you guys, that's actually fairly difficult to contract HIV. We'll learn this later. It's not like if you had a sexual encounter with somebody who's HIV positive once, your odds are fairly low of actually contracting it. It requires many exposures. So we'll come back to that later, though. Now, what about these cytotoxic T cells? What was their function?
They are killers.
The Grim Reapers, right? They're the killers. So when they touch a foreign cell, what does it cause that cell to do? Die through a process of programmed cell death we call apoptosis. So that's what you're seeing here, you guys. Right when that cytotoxic T cell touches the foreign cell, it goes, ah! And it starts to wither and like start to die, right? So it's not cool. Like this is happening in your body right now. I think that's cool. But here's the caveat, you guys. These cytotoxic T cells are only activated with cytokines. So you need T helper cells to release cytokines in order for these cytotoxic T cells to also be activated. So what does this mean then for people with HIV who have less T helper cells and therefore aren't releasing as many cytokines? Well, I'll just answer myself. They have a lot of cytotoxic T cells, but these cytotoxic T cells are basically ineffective, right? Because they're not receiving the signal from T helper cells in order to actually participate in that infection. And that's sort of the insidious thing with HIV is that, you know, there's a lot of immune cells present, but they're not able to participate in the immune response as well because there are aspects of your immune system that are now sort of taken out. And it's just sort of like like the Achilles heel of your immune system. You remove those T helper cells, and although you might have a whole body intact, like that's sort of a big enough wound to where the rest of your immune cell system can't really participate in fighting that infection as well. So it's kind of interesting. Now, the B lymphocytes, like the T lymphocytes, are part of your specific or adaptive immune response. So they only respond to one type of antigen. So what this means, you guys, like the T cells, they only respond to one type of antigen. If you've ever been exposed to like influenza virus or chickenpox virus or had like rhinovirus, which is like the seasonal cold, then you're going to have B cells and T cells that respond to antigens that are specific to those microorganisms. But what this means then is that if you're ever re-exposed again, then you'll have these cells present to respond to that specific antigen, like on influenza. So let's say if you had another flu in like 6 years, like last time you had the flu was back in 2010. And who knows what we were doing back then? But let's say if you're exposed to influenza again more recently, well, you already have cells in your immune system that can respond to the antigens on influenza virus because they go dormant. Now it includes B cells and T cells respond to one type of antigen. What this means, though, is that if there's lots of different types of microorganisms out there, that means we need a lot of different types of B cells and T cells to respond to those microorganisms. But that's why they're specific, is they only respond to one antigen, not multiple. Yeah?
What happens if viruses mutate? Are
Exactly.
they still able to respond?
You got it. That's why influenza, and that's sort of why you mentioned that one. That's why influenza can reinfect you, right? Because it mutates so rapidly from season to season that it has new antigens. And those antigens are different enough to where your existing immune cells may not recognize that new antigen, and then that's when the virus can kind of take hold. And influenza is particularly interesting too because it actually has chromosomal structure like genomes. And this virus actually, it's not really appropriate to call it like sexual reproduction, but when multiple viruses infect a cell at the same time, they shuffle their chromosomes. And when they shuffle the genetic code, that means that their progeny are like a shuffled deck of cards. They're slightly different. They're similar, but also slightly different than what they came from. And that's why influenza can mutate so fast. And it's an RNA virus. So its mutation rate's like 1 in 1,000 base pairs. But we'll come back to that too. But you were definitely raising an excellent point. This is also why in some people who have HIV, even if they have B cells and T cells against HIV, HIV also mutates within your own body. So it's kind of interesting, and that's evolution, you guys. Microorganisms change; they evolve. That's what it is. Evolution is just the process of change, right? And you can see this occurring season to season, or even within someone's body. So what's cool though about these B lymphocytes is that they only respond to one receptor, right, or only one antigen. Okay, that's maybe not so cool, but they can specifically fight a particular infectious agent or antigen. And they do this by creating antibodies. So antibodies, also called immunoglobulins, remember this is gamma globulin, you find in plasma. So we have alpha, beta, gamma. So gamma globulin were antibodies, and it's a type of protein that can stick onto foreign antigens, right? Let's say if you have type O blood and you're exposed to like A antigen, then you'll start to make antibodies against that A antigen. So what this means is you'll have B lymphocytes that are making antigen or antibodies against that foreign antigen. Okay, and they start pumping out these little antibodies. Now they're also sensitive to cytokines as well, you guys. So cytokines can help activate these B cells. And what this next slide shows are some of the functions of antibodies. So antibodies can help stick onto foreign microorganisms and neutralize them. Especially in the case of viruses. For viruses, they need to get inside of a host cell to replicate. Like viruses don't have organelles. So they can't just divide on their own. They got to get inside of a host cell, then use the host cell's organelle machinery to replicate itself. Okay? But if you coat that virus with an antibody, like let's say if you have a little virus here, and you just start coating it with antibodies, then it can't get inside of a host cell. Right? You're sort of like putting a ball and chain on it, and it can't move inside of a host cell, so now you neutralize it, and now it can't actually be infectious. Antibodies are also involved in agglutination. So we talked about how you can get agglutination reactions with improper blood transfusions. But this is a normal consequence of antibody function, where antibodies will stick onto a foreign antigen and start causing these cells to start to clump together. What's the function of this? Why would you want to have cells clump together like a little local area? So they're all in one spot, which makes it easier to, you got it, makes it easier to remove them, right? You kind of start wrangling them up. You got all these foreign cells in a little area, and if they're loose, it's harder to catch them all, right? Well, think of the antibodies here in terms of agglutination. It's kind of wrangling these things together. It gets into in a nice small little area by sticking them together, and it makes them then easier to remove by your immune cells. Okay. Now, antibodies are also involved with precipitation. Precipitation means that it can take things out of solution. So an example of precipitation, you guys, is like where like sugar can precipitate out of water and form little crystals, get like little sugar crystals, right? Well, one thing that can help initiate precipitation in your body are these antibodies. So what if you have a toxin in your body? Well, an antibody can stick to it and precipitate it and make it less dissolvable in water. And if it's less dissolvable, then it's easier to remove. That way it's not just floating around. Antibodies also activate complement. This is crazy, you guys. Complement, to me, is one of the coolest things. Complement are a series of proteins that are made by your liver, okay, as well as other immune cells. These complement proteins are part of the clotting cascade and fibrinolysis, so clot breakdown. But they're also part of your immune response because when antibodies bind to a foreign antigen, it activates complement, and all these proteins start to stick together into a tube-like structure. And once you form this little barrel structure in the foreign cell, it's basically like punching a hole in that foreign cell. So these complement proteins actually form what they call a Mac-attack complex right here. It's a little barrel of proteins. And when the antibody binds the foreign cell, all of a sudden these proteins start going, and they start lining up in a barrel. It punches a hole in a foreign cell, and then guess what happens to that cell? It lyses. It explodes right here, like the Death Star. Right? Just look at this. So you get this little Mac-attack complex, which is formed after an antibody binds to a foreign cell. It punches a hole in that cell, and it causes the whole cell to explode due to an influx in water. So the whole cell starts to swell up and explode. That's pretty cool. Now, have you guys ever heard of like Anti-Venom?
Yeah.
So, like let's say if you went for a hike. By the way, I've never seen a rattlesnake in Colorado. Apparently they're here, right? I grew up in a desert. You've seen them out here? I grew up in a desert, and they're all over the place. Like you can't really go out in nature without seeing one. But here, like I've been here for 11 years and I hike a lot. I still haven't seen a freaking rattlesnake. And I don't know if it's because like I'm not looking, or they're hiding, or maybe they're just maybe a little bit of both.
Above 9,000 feet because they don't live above 9,000.
A little bit of both. A lot of times just like local area.
Golden, if you go in the spring up to the Golden Trails,
Yeah.
the ones that face the sun.
Oh yeah,
during the
like
day,
Apex Park.
Yep.
Yeah.
you will
Someday.
So let's say if you're on a hike, you get bit by a rattlesnake, right? You're just like, oh, I'm enjoying my little hike, and then all of a sudden, next thing you know, you have this like weird slithering creature stuck into your thigh, or right in your calf or something, right? And you can see it like injecting venom into your leg. You're like, oh no. Right? Oh dear. What do you do now? Right? You just lay there and just like contemplate your life, like, well, I had a good run. And then use like your remaining minutes to just kind of think about your life and reflect as your like cardiovascular system starts to shut down. No, right? You go to the hospital and they give you anti-venom. What if I told you guys that anti-venom are antibodies? But they're not antibodies from humans. They're antibodies from animals. So you know when they milk the snakes to get anti-venom? What they're doing is they get the little snake and they get to bite the cup and then gets its venom in a cup and they collect it and they might get like 6 drops and like, oh, that's a good milk, right? What they do is they take that venom, they dilute it out, and then they inject it into animals like rabbits and rats and goats and sheep. But those venom that you find, it's diluted out. It's not enough to kill these animals, but it's enough to stimulate an immune response in those animals. It causes a goat or a rabbit or a sheep or a rat to start making antibodies against that foreign antigen, which is the venom. What scientists can do then is extract those antibodies out from the goat, rabbit, rat, then use those antibodies and inject those into somebody who's now been bitten by a rattlesnake. Because now what they can do then is take a goat antibody, put it in your bloodstream, and then that goat antibody can stick onto the venom and it causes that venom to agglutinate or precipitate or neutralize. And so that way that venom doesn't have an effect on your body. So it's actually pretty cool. However, though, you guys, you couldn't get that too many times because you can start to make antibodies against those foreign antibodies. If that ever happened, then you would be kind of SOL, out of luck, because now you can't even use those antibodies for anti-venom. But for people who have been bitten by snakes a lot, they start to make their own antibodies. So you hear about like immunity, like there's a snake charmers and stuff who like they can get bit by cobra and then like they don't even really get much of an effect. They might get like a little bit of fever, you know, maybe some bad dreams that night.
But they don't die.
They don't die, exactly. Yeah, because they've been bitten so many times that their own immune system has antibodies against like cobra venom. I know, so that's pretty cool. But how does it work as an antivenom? Well, it can neutralize that, cause it to agglutinate, or even precipitate, which is actually pretty cool. Now, for the B lymphocytes, we talk about them as being like the antibody factories, right? Now, the ones that are actually making antibodies are called plasma cells. And these plasma cells make antibodies at a tremendous rate, and an enormous rate once they're activated. These antibodies they get exocytosed out of the cell, and they float around your body's solutions. Think of like your blood plasma, interstitial fluid, lymph, even joint fluid, and then those antibodies then can go on and do their thing, right? They can agglutinate, they can neutralize, they can precipitate, or even activate complement, which can punch a hole in a foreign cell. Now, some of those cells, though, go dormant and they form a memory of that infection, because not all of these cells participate in the immune response. Some of these clones that develop, they just kind of hang back, right? They go, okay, you guys make antibodies. I'm going to hang back and just remember this for a later date. I'm going to remember this for one day, right? Whenever it comes back. So that these memory cells go dormant in your lymphoid tissue, like think your lymph nodes, your tonsils, your bone marrow. And these memory cells, they store a memory of that infection. That way, if you're ever exposed to that antigen again, you got memory cells ready to go. And you can initiate an immune response much more quickly. So what does this look like here? Well, when cytokines are released by T helper cells, they can activate a little B lymphocyte here. And these B lymphocytes can divide into clones. You see that not all the clones are the same though, right? Some of these form plasma cells, which are larger and they make antibodies. Some of them actually turn into a memory cell. And the function of a memory cell is they actually are senescent or sort of quiescent. They're kind of more dormant, but they can reactivate if you're ever exposed to that antigen again. So, and this can last decades, if not entire lifetime. It's kind of cool. Now, these plasma cells make antibodies, which participate in the immune response. However, these plasma cells their longevity is not as long as like a memory cell. So these plasma cells might actually die after the infection is over with, whereas a memory cell will just go dormant in a tissue somewhere. And if that infection ever comes back, or if you're ever re-exposed to that antigen, then these memory cells can kind of kick in. So what does this mean if you've ever been bitten by a rattlesnake? So you got bit by a rattlesnake.
The first time's going to be the worst.
The first time's going to be bad, right? Because now you don't have immune cells to make antibodies against the toxin. Let's say if you survive, though. And let's say from that bite, you generated memory B cells against rattlesnake venom, right? What happens the next time you get bit by the rattlesnake?
It's not going to be bad.
Memory B cells will activate, which we see here, right? And then they're going to make more clones. In fact, you see that the second response is not even, it also occurs more quickly, but it's also an even greater immune response. You get even more plasma cells and even more memory cells from that second bite. What about a third bite?
It'll be okay.
Yeah, well, you'll
You
get
got even more, right?
Plasma
Now you have
cells and
more
more
memory
memory
cells.
cells.
Yeah, which means the third time you get bitten, you'll probably have even more plasma cells and more memory cells. So you start generating more and more immunity to this. And just on the whole idea of snake bites, you guys, I can send you the video. It's kind of weird. But there's this guy, and he's an American guy living in England. But his thing is he's got like a collection of the most deadly snakes on Earth in his apartment. And he keeps them in these little, what do you call that? Little,
Terrarium.
not a terrarium. It's even worse than that.
Big boxes.
Yeah, like a box. Like a
Like a Tupperware.
Tupperware box. He keeps them in Tupperware, okay? He'll pull them out occasionally. He milks them, right? And then he dilutes out their venom and then injects himself with the venom. And like his thing is he thinks like he gets high off of this stuff. And for whatever reason, he's trying to develop immunity against the venom. Someone actually looked at how much venom he can now inject into his body. It would be enough to kill like an elephant. In this documentary, you see him inject it into his own body, and he's totally fine. He said he like got a little sweaty and like looked a little uncomfortable, but that was about it, you guys. So what this means then is that after so many exposures, he's ultimately about as immune as you can get to this venom, right? So it almost has no effect on you. It's the same amount of cobra venom that will like take down an elephant. This guy, just like a normal sized guy, no problem.
Moral of the story: if you can get past your first one, you'll
Yeah,
be fine.
exactly. Yeah, get past the first one, you'll be fine. So generate a stronger response.
Living in like a region where there's a lot of snakes, why don't they just do like preventative exposure for people that live in really snake-ridden areas?
That's a good question. Good question.
It would be like a snake venom.
Yeah. Yeah, exactly. Yeah, exactly. And you could just sort of just inject yourself with a very dilute version of that toxin.
Just over and over again. So
Yeah.
if you ever got bit on a trail, you're good to go.
Exactly. I know, right?
That sounds like a lot more.
Something the military would do.
That's something the military would definitely do, right? Especially if you're like,
They
I don't
give
know,
us
like jungle
anthrax
combat or
vaccines.
something. Yeah, you guys get vaccines that not everyone gets. Now, what this slide has shown you guys is the primary versus the secondary immune response. So let's just run with the whole venom thing. Why not? I mean, it could also be an infectious microorganism. But let's say the first time you're exposed to that snake venom, you're not going to generate an immune response for up to a week. Okay. In fact, let's get away from snake venom. Actually, it's a little confusing. Let's just say influenza. Let's say if you're a child, you've never been exposed to influenza before. You got a totally just sort of naive immune system against influenza. You're first exposed, day 0. You won't even see antibodies being created until close to a week later. Okay? You'll start to generate antibodies against that influenza antigen, right? Once the infection goes away, then antibody titer will start to decrease over time, right? But let's say sometime later, you're re-exposed to influenza again, and it's the same version, so the same antigens. You see the second response occurs almost immediately, and you get a huge and rapid increase in antibody titer very quickly, right? That's a secondary response. Now, this is also a good slide to talk about the whole idea, like the rationale behind a vaccine. In fact, you guys see this between B and the second response? This is showing what your immune response would look like if you weren't ever exposed versus after being exposed prior. So you can see it's more rapid and a stronger response the second time. But this is also a good time to talk about like how vaccines work. So the vaccine, you're exposing yourself to antigens early on, like before an actual infection, right? Like they have an anthrax vaccine. And military personnel receive this. It's not like for the whole population, which I don't know why. Maybe there's the risks of it are too high or something.
Postal carriers get it too.
Yeah. But let's say if you get the anthrax vaccine, right? What are you getting? Well, you're getting antigens from Bacillus anthracis. But you're not actually getting an infection because you're not actually being exposed to like an infectious microorganism. After some time, you start to generate an immune response against those antigens, and then the kind of response dips away over time. What happens if you were ever exposed to anthrax, though, after getting the vaccine? Yeah, it's like getting a secondary response without ever being infected to begin with. So you can stimulate a primary immune response without ever having to be infected. So that way, actually, when you are infected, or if you do get infected, your secondary response occurs more quickly and is more dramatic. That's the rationale behind vaccination. Or just think about like chickenpox vaccine, same thing. But what about memory B cells? Do they last forever?
Yeah, they
They
can.
can, but the thing about cells in your body, though, is they all have a lifespan, right? So let's say if it's been 10 years since you've had the vaccine, you probably have less memory cells than as you did, the 2 weeks after you get the vaccine. So what this means is this is the whole rationale behind booster shots, is you're trying to stimulate an immune response again to generate more memory cells. That way, if you're ever re-exposed, then you'll generate an effective immune response.
Like tetanus would be that way.
Yeah.
That one every 10 years.
Yeah, you need a booster. Yeah.
So